Hello, my name is Laura Skurlos, and I am a senior scientist at Sartorius. In this video, I will be presenting research conducted by our team at Sartorius, including Susan Fulton, Hina Kempwala, Eric Black, Aslam Dagani, Nicola Bevan, and Cicely Schramm. Note that this research was presented earlier this year at the International Society for Cell and Gene Therapy 2024 conference in Vancouver, Canada. Our poster, entitled Real-Time EV Uptake Characterization Using an IncuCyte Live Cell Imaging System, describes an innovative labeling approach that allows continuous monitoring and analysis of EV interactions with cells. Over the next few minutes, I will walk you through the methodology, validation, and key findings of our study. Extracellular vesicles, or EVs, are small particles released by cells that play a crucial role in intracellular communication. They carry proteins, lipids, and nucleic acids, which can influence the behavior of recipient cells. Understanding how EVs are taken up by cells in real time can provide valuable insights into their functions and potential therapeutic applications. Here we describe the use of a covalently bound dye optimized to label EVs for real-time analysis in the IncuCyte Live Cell Imaging System. The IncuCyte Exaflor Green EV Labeling Kit provides a novel method for the real-time visualization and quantification of extracellular vesicles. By employing a covalently bound dye, the kit offers a stable and uniform labeling of EVs, representing a significant improvement over traditional labeling techniques and reducing background noise. The kit's compatibility with a variety of EV purification methods and cell origins ensures its broad applicability in live cell assays, facilitating diverse research opportunities. The Exaflor Green labeling dye enables EV-specific visual tracking of EV uptake, allowing for precise studies of EV dynamics without the interference of nonspecific dye attachment. Researchers can leverage the kit to investigate EV uptake and functions such as wound healing to enable a deeper understanding of intracellular communication. Mesenchymal stem cell, MSC-derived EVs, labeled with Exaflor Green, demonstrate uptake and kinetic migration effects using the IncuCyte scratch wound cell migration assay. To begin, let's walk through the Exaflor Green labeling method for EV uptake characterization. This mix and read method involves several key steps. First, seeding the cells and allowing them to adhere to the plate overnight, followed by EV labeling using the Exaflor dye, which is an 80 micromolar reaction at 37 degrees for 30 minutes. Next is the free dye removal using the VivaSpin 2 ultrafiltration column, and that includes a spin at 2000 G for 5 minutes, followed by a cleanup step using the MiniSART filter, a 0.22 micron PES filter, which sterilizes the sample and removes aggregates. In the following steps, the media is removed and replaced with the media containing the labeled EVs. The kit includes both the Exaflor labeling dye along with the consumables necessary for quick and efficient processing. The protocol, as described, ensures uniform labeling of the EVs and removal of the free dye to prevent the nonspecific background fluorescence. In order to generate EVs, human bone marrow-derived MSC cells were cultured in a 3D Sartorius glass univessel bioreactor for 5 days using Nutristem XF MSC medium with supplements and human platelet lysate, or PLT. Post-culture, cell aggregates were washed with DPBS to remove PLT and incubated for 3 more days in fresh medium to secrete EVs. The conditioned medium was then processed to purify EVs, clarified via SartoPure PP3 filters, concentrated with a SartoFlow Smart System, treated with nuclease to remove DNA, and diafiltered for ion exchange chromatography. EVs were isolated using a SIM-Multus IEX monolith pooled from fractions and underwent a second tangential flow filtration, or TFF, for buffer exchange. Final EV concentration and purification were achieved with a SartoPore 2 XLG filter. Quality and concentration of EVs were assessed using protein analysis, nanoparticle tracking, Western blotting for particle markers, and fluorescent lipid SEC with PatFix HPLC. Validation of the EV labeling kit was performed using A549 Hansa Biomed exosomes and PC3 cells. The data presented here demonstrate the data analysis tools to measure the successful uptake of labeled EVs by these cells, confirming the compatibility and effectiveness of the labeling kit. Figure 3A shows the average green object mean intensity increased as a measure of the A549 cells taking up labeled PC3 exosomes in a dose-dependent manner. Note also the dye-only and medium controls show no fluorescent signal. In Figure 3B, histogram analysis is used to classify populations of segmented cells at 24 hours. Note controls clearly gated as low or high green population cells. The gated populations of cells plotted over time is shown in Figure 3C, again noting clear differentiation between the controls and the labeled populations. Figures 4A and B demonstrate the kit compatibility across a panel of adherent cell types at 24 hours post-treatment. A549 Hansa Biomed exosomes were labeled using the IncuCyte ExaFluor dye, and uptake was observed alongside control wells. The top row of images show intracellular fluorescent signal of labeled EVs. The bottom row of images are the dye-only control wells for each condition. These images verify the fluorescent signal is not due to the residual free dye after processing. Phase images also show no change in cell morphology compared with controls. In Figure 4B, plots of the object count normalized to time 0 illustrate the uptake of labeled EVs does not cause perturbation. An important aspect in developing this kit was to provide a labeling method that is broadly applicable and not limited to a specific type of EV. It is well known that production and purification methods can significantly affect the purity of EVs. This figure demonstrates the labeling and uptake of low and high purity EVs labeled with IncuCyte ExaFluor Green by A549 cells. Images shown here are taken at 24 hours post-treatment. Shown are human plasma-derived exosomes purchased from Abcam and purified using ultrafiltration on the low end of the purity spectrum. A549 exosomes purchased from Hansa and purified using ultrafiltration and SCC are also shown, and followed by the Sartorius production lots of MSC and HEC293T EVs extracted using TFF ion exchange chromatography. Images demonstrate the robustness of the kit across various EV production methods. In Figure 5, we explore the concentration dependence of EV uptake. The data and images illustrate how the uptake of labeled EVs varies with concentration, highlighting the importance of optimizing EV concentrations in experimental setups. Labeled EVs were purified from HEC293T cells, an in-house production lot, and titrated about 40-fold from 4 E8 to 1 E7 particles per well. Phase and green fluorescent images were acquired every 2 hours over the course of 48 hours, and uptake was assessed using the average green mean intensity. These data are shown in Figure 5A. Figure 5B shows the phase object count normalized to time 0, and the data here illustrate no change in proliferation at all, concentrations of EVs tested. The aim of Figure 6 is to show mechanism of action for exosome uptake, specifically focusing on endocytosis. The data and images show the concentration-dependent inhibition of EV uptake by Dinosaur in A549 cells, providing insights into the pathways involved in EV internalization. Dinosaur is a known inhibitor of dynamin, a GTPase involved in clathrin-mediated endocytosis. Figure 6A illustrates that using this inhibitor inhibits the uptake of labeled EVs. Figure 6B shows the concentration response curve generated from 24-hour time point data, reflecting the expected drop in green object mean intensity with an IC50 of 2.72 micromolar. Representative images, also at the 24-hour time point, provide visualization of Dinosaur inhibition of EV uptake at the indicated concentrations. Figure 7 presents the scratch wound migration assay supported with the IncuCyte. This assay allows us to study the impact of EVs on cell migration, which is a key process in wound healing and cancer metastasis. By creating a scratch or a gap in the confluent cell monolayer, we can monitor the migration of cells into the wounded area over time. The IncuCyte system provides real-time imaging analysis, enabling us to quantify the rate of wound closure and compare the effects of different EV treatments. The scratch wound migration assay involves 4 steps. The first is coating with an ECM, or an extracellular matrix, and in this experiment we used collagen. Next is seeding the cells at a density of 10 to 40,000 cells per well. In this experiment, NHDF cells were seeded at a density of 15,000 cells per well and plated overnight, creating the monolayer. On the next day, the wound area was created using the wounder, and finally, the treatment was added, in this case, using the complete medium DMEM plus 10% FBS, assay medium, which is a serum-free DMEM, or assay medium containing the MSC-derived EVs labeled at a concentration of 4.5 E9 particles per well. Cell migration was captured using the IncuCyte live cell analysis software system, and relative wound density, quantified over time, is shown in Figure 7b. When labeled EVs were added to NHDF cells, there was a significant increase in the relative wound density compared with cells in wells treated with the assay medium alone. In this case, the difference was 81% versus 65% at 72 hours. As expected, the NHDF cells grown in complete medium, which was DMEM with the 10% FBS, achieved a relative wound density greater than 90% in the first 24 hours post-scratch. Phase contrast image analysis, shown in Figure 7c at 18 hours post-scratch, highlight the variation in cell migration across the treatment conditions. Initial wound boundary is mapped for reference. Notably, the cell morphology in complete medium is comparable to the cells growing in optimized assay conditions. In conclusion, the IncuCyte Exaflor Green EV labeling kit enables real-time visualization and quantification of EV uptake in live cells with a stable, covalently bound dye. Validated across various EV purification methods and cell types, the kit facilitates the study of EV uptake mechanisms and their functional roles, such as in wound healing. The kit's specificity allows for accurate EV tracking in live cell assays, supporting standardized research into EV dynamics and functions.